Hi, and welcome to Experience Magazine Live. I'm Joanna Weiss, the editor of Experience, and I am joined by Matt Crossman, our adventure and fishing correspondent, and by Dan Distel, who is director of the Ocean Genome Legacy Project at Northeastern University's Marine Science Center in Nahant, Massachusetts. And we are here to talk about Matt's recent story for us, which has a kind of cryptic title. Did a legendary trout really ride the rails from California to Missouri? And I encourage you to read it because it's an amazing story about fish and DNA and the power of naming a living species. And Matt, I am going to just hand it off to you to talk about it a little bit more.
Well, thank you. Fishing correspondent, I'll add that to my resume. That's a new one. I joke with my fishing buddy that because I get to catch fish, I'm actually now a professional fisherman. And he's seen me fishing. That's a great big load of hooey. But yeah, the story that I wrote was about chasing an elusive rainbow trout that lives, perhaps, we're not really sure, only in one particular creek in Missouri. And in researching that, I talked with Dan Distel, who joins us here. He's the director of the Ocean Genome Legacy Project, and I will have him define that a little. But my first question is just about the broadness of the DNA research, both eDNA, which is environmental DNA, and otherwise that you guys have uncovered isn't the right word, but that you preserve in the ocean. What are you guys doing there? What is your mission?
Yeah, so basically, you know that the ocean is full of amazing creatures, all kinds of species, most of which have not been yet described or are not yet understood. But then again, many that are extremely important to us, like the seafood species that we all know and love to eat. And we also know that we're having a big effect on the environment, and by that I mean mankind. And as a result, a lot of species are disappearing. So part of the mission at Ocean Genome Legacy is to capture the DNA from those organisms, because that contains a lot of information about each one of those species. And then we freeze it away in our freezers, and then we make it freely available to researchers all around the world who can use it for any sort of research that they like. And that can range from things like medical research to agricultural research, food-based research, all kinds of different types of research are being done with samples from our collection.
Do you have, how many samples do you have? And is there any way to even guess how many are yet out there?
We have in the neighborhood of about 27,000, I think, or so samples, DNA samples in the collection. And that's close to 5,000 species represented there. Now, what's in the environment that hasn't been discovered? That's a great question. It's estimated that the marine environment could contain at least 2 million species, possibly many more. And to put that in context, so far the number of named species on Earth is just under 2 million. So, and the number of named species in the ocean, that is the ones we know about and we have names for, is around 240 to 250,000.
Wow. So you're never going to run out of work to do.
No, we're going to keep busy.
Why don't you show us around the office there?
We are a pretty modest operation. We have a small footprint, but I would say what we managed to do in this small footprint can have a big impact. So I'm going to swing around and take you to where the collection actually lives. And so surprisingly, DNA samples are very small, so we can keep a lot of samples in very small space. And so this is where the freezers are. But each of these freezers, you can see, is very, very cold. I don't know if you can read that, but that's at minus 84 degrees centigrade right now. So that is a very, very cold temperature. And at that temperature, we can keep samples essentially indefinitely without any degradation. We've named them after famous scientists. So this freezer is Linnaeus, and this one is Darwin, and this one is Franklin. So those freezers are just like the freezers you have at home, just much, much colder. This is a liquid nitrogen freezer. And I'm just gonna give you an idea of what that looks like. So that is storing samples in liquid nitrogen, and it's currently, as you can see, I don't know if that's readable, minus 182 degrees. So that's really cold stuff. And in addition to that, here you can see we have these explosion-proof storage containers, and that's because we store various kinds of samples. These are what are known as voucher specimens. And this is what you'll see in almost any museum. For example, here, I'm not sure if you'll be able to see that. That is a starfish. These are samples in ethanol. We call them voucher samples. And what they do is, if there's ever a question as to the identity of any sample in our collection, we have these voucher specimens, which is, you know, a whole specimen of the animal, which can be used to check out the morphology or the anatomy.
One of the things, I mean, almost everything in that story that I wrote surprised me because I didn't have a grasp of the vastness of DNA research. But one of the things that surprised me was the fact that you could be looking at two animals. There was a butterfly that was an example, and a fish called a sculpin that was another example, that even a butterfly expert or a fish expert could be looking right at it, and you can't, they're different species and you can't even tell. So that makes the possibility of the vastness of species that extrapolates that out to a number we couldn't even guess. And you and I had
greater. That's right.
That the more questions we ask, the more we find out, which just reveals we know even less than we thought in the first place, you know?
Yeah, that is true. We have many examples of species that we can't tell apart. We presume they can tell each other apart.
Huh.
But
on that for a second. So you're telling me like that, let's use the sculpin and the prickly-like sculpin as an example. Just, it doesn't have to be literally true, but the prickly-like sculpin could swim up to the other sculpin. I'm like, no, you ain't one of us.
Pretty much, yeah. A lot of that is through, you know, it can be through very subtle cues, you know, very subtle differences that they can detect, but also can be through chemical cues. You know, there are chemicals, for example, called pheromones that enable organisms to recognize potential mates and help them to stimulate reproduction. But yeah, we know that they can tell each other apart because they do. Very often we have cryptic species where even the territories, the regions where they're found overlap, but because they're distinct species, we know that they don't reproduce with one another. So, but we have examples. For example, I work a lot with a group of organisms called shipworms. They're bivalves that eat wood, and the old sailors in the old days used to be very afraid of them because they would rot the hulls of their ships very quickly. But we have certain species that we cannot tell apart until we put them in the blender and extract the DNA and sequence the DNA, and then it's very obvious that they're different. But by eye, we can't tell the difference.
Is there any, I mean, I'm not sure how to ask this question. Do we understand what we don't know? Like, are we missing something? Are we not looking at it correctly, do we think? Or is it just, there's a difference there that we just can't see?
Probably a little bit of all of the above. So in many cases, there's differences that are hard for us to see. And it's because we don't know what to look for. So that's where the unknowns come in. The more we study, the more we understand what it is to look for. For example, we only see a portion of the visible spectrum of light. there are other organisms that see different parts of that visual spectrum. And very frequently, scientists are finding, for example, in the oceans and in the deep sea where it's often dark, scientists will go down there and shine different wavelengths of light and suddenly realize that, hey, some of these things that look very nondescript are now glowing with very intricate patterns. And those patterns must mean something to their mates or to their predators or to their prey. They're not there for no reason.
That must be just fascinating to go down there and think that we observe and absorb and measure and collate the world in a very particular way. And we just assume that all other animals do that. I mean, I guess, at least I do, assume that they do it the same way. And it sounds like that, not only is that not true, but there's ways that they do it that we don't even know or understand.
That's right. And it's only by comparing things. And, that's one of the great things about biodiversity that people may not realize is the way we really learn about species is by comparing different species. For example, if you have two different organisms that have solved a particular problem of how to live, say, at very cold temperature, or how to live in very dark or at high pressure, all these, common things in the oceans. Different species have learned different ways to solve that problem. And when you begin to compare them, you begin to really understand what's the basic chemistry and physics and the basic biology that explains how they do it. Sometimes I like to say, if a Martian came to Earth and saw an automobile, right, a car, have no idea what that was or what it was for. But the more cars you look at, you begin to see, oh, well, they all have four wheels. That must do something, they all have an engine. That must do something. And by comparison, you begin to figure it out.
What has that taught you about the power of life, if I could put it that way, that in order for a creature to live way down at the bottom of the ocean and for us to be so completely different, there's something powerful going on there. And I'm wondering what if you've drawn any conclusions or what you think about that.
Really, there's so much that we don't know, but it's just beyond our reach. We can every day find something new and something unexpected. So much so, for example, I mentioned the shipworms before. Yeah, in the old days, they were very important to the British Navy. There's reams of information written by the British Navy about shipworms. But also, we found more recently that shipworms have bacteria that live inside their cells, actually inside their cells, like an infection. Some of those bacteria help them to digest wood. They produce the enzymes that help them to digest wood. But recently, we found out that they also produce antibiotics. And so who would have thought of looking inside a clam in the ocean that eats wood to find a potential medicine? We would never think of it. But by accident, by studying other things, we stumble across it. And it turns out we have colleagues in Utah and now in Minnesota who are working on various potential candidate drugs that come from these bacteria living in these organisms. And they may have a big effect on people's lives.
All right, the more practical question, if I'm out fishing either at Crane Creek or somewhere else, and if I catch something that I think has never been seen before, how can I contribute? How can just a regular fisherman contribute to all this?
See, we love to hear from people to tell us, hey, we found this crazy thing. Because sometimes it's something very unusual and they can help us to obtain samples of it. Sometimes it's just something common, but so spectacular that people really need to know about it. And we put things like that in our newsletter or in the Northeastern News. So we'd love to hear from people.
Thank you guys both so much for a fascinating and profound conversation, just like the story. I'm not going to think about fishing or freezers again exactly the same. And anyone who wants to read Matt's story or any other story from Experience Magazine can go to expmag.com